Hey everybody, this is a one of the problems that came up during lecture today, and it seemed like there is some confusion on it, so I'm just making this video to try to clear things up the best I can. So this is the picture. We're going to go off to this side and talk about the different types of transporters there is, and then we'll come back to this picture. So in transporters, we have active transportation, and then we have diffusion. So in active transportation, we have primary and secondary. So your primary active transporters are going to be things that use ATP or maybe GTP, and a lot of the times they'll be called pumps. So if we go over to this picture down here, we can see this has ATP and it's a pump. So we know that that's primary. So we'll mark that as, we will mark this as primary for now. And let's talk about secondary. So secondary uses the momentum of one molecule to pull another molecule through. And so this is going to be your antiporter and symporter. I memorize these by sim, sounds like same. So symporter is bringing them the same way. Anti is anti, like one's against the other, and that's how I memorize those. So we'll do S and A. S is for symporter, A is for antiporter. And 1 second, I'm just drawing this picture. So there's a lot of blue up here. There's a lot of blue up here. So, and there's a lot of red here. There's a lot of red up here. Alright, so you can see in the symporter, we have this red molecule going with its concentration gradient, how it should, and the momentum created from this is enough to push this blue one against its concentration gradient. It's going from low to high. Over here, we have this red molecule with its concentration gradient going from high to low, and it creates so much force when it goes from its high to low that it's able to bring this blue from low to high. And that's why it's active. So we can, a good rule of thumb is active is low to high. Diffusion is high to low. Now with diffusion, you just have regular diffusion, which is called passive. And then you have facilitated, usually by a protein. So passive diffusion, I like to think of like air in a balloon. Like let's say you blow one balloon up really full and then another one like halfway and you connect them. They're going to balance out the air between them. So that's a good way to memorize passive. Facilitated, you want to think about like an aquaporin channel, which we mentioned today. Maybe a alpha helix. There's going to be some sort of protein that is holding a gap open for them to come through. All right, so now that we know our active and diffusion, we're going to go over here and we're going to solve this problem. So we know glucose is high in the middle. Let's zoom in some. So we know glucose is high in the middle. And we can also tell, even if this wasn't here, we could tell that glucose is high in the middle because this is a glucose passive transporter. So that means glucose coming through here is going to go from high to low. So because this arrow is going this way, we know that this is high and this is low. We also know that because this is a sodium-potassium pump, the Na concentration gradient is higher here. And that's why we need a pump, is to actually push that sodium into the bloodstream. Okay? So now we know that sodium concentration is lower in the cell. Okay, so this is kind of starting to make sense that it's going to be higher here since glucose is high. So we're pushing this sodium is going from high concentration to low concentration, and it's using that energy to pull in this glucose molecule from low to high concentration. And that makes sense. This would be our symporter. So this is an example of symporter. Okay, cool. So now we got the ATPase pump, which is pumping sodium from a low concentration to a high concentration. Because remember, this pump, if we go over here, is low to high. And what we're seeing here is this one. Alright, and then over here, this passive is what we see here. Alright, so I got squiggly lines going all over the place. So when you go down to here, the glucose, we know, what are we doing? Where is it highest? So we know glucose is highest in the enterocyte, so it's highest in enterocyte, lowest in bloodstream, and lumen. We know that our sodium potassium is going to be higher in the lumen and lowest in the enterocyte, lumen and blood. And the potassium, we know it's highest in the cell and lowest in the bloodstream. Okay, so hopefully this really cleared up why these things are working how they are, what's going on. There's a lot to look for in this picture, so let's go through it. I'm going to go through it one more time and just kind of spot out the main things you guys need to realize when you see something like this. Okay, so we're going to pretend that we just don't even have this. We just don't even have this little tip for us. We're going to pretend we have no idea what's going on. Alright, so we're going to look straight away. We're going to read all of these. Just go through, read all of these, familiarize yourself with this and understand what's going on. Okay, so let's look at our big things that just give it away. So we have passive transportation. Glucose is passively transporting from here to here. So that means this must be low concentration. This must be high. Over here we have a pump, which makes things go from low to high. Okay, so Na must be high here, low here. K must be high here, low here. If glucose is high in the cell, but it's low outside, or you could work it from glucose or you could work it from sodium. So let's go to sodium. We'll say that sodium is low and this one is high. Okay, right off the bat, we know this is following its concentration gradient. And since it's a symporter, that means the other one must not be following its gradient. Because symporter brings one from low to high and one from high to low. So this one must be low to high. And definitely try to understand this relationship right there. Understand this relationship with symporters, that one goes low to high and the other goes, whoa, what am I writing? The other goes high to low at the same time. Okay, so memorize that, and this is symporter. Antiporter is going to be the opposite, obviously. So hopefully this helps you guys out, and that's it for today's video.